Hey everybody, so we're going to take a quick look at two different kinds of Punnett squares. These are two examples of what we call non-Mendelian inheritance, meaning that Gregor Mendel didn't think of these ones. So these are kind of were discovered or identified after what Gregor Mendel did, and he didn't know about these when he was doing all of his experiments and stuff. But it came afterwards. People who studied this more in depth found out all of this information. But basically, when he was doing his pea plants, he was able to identify only just really two different alleles, tall, short, or pink or white, or number of leaves on the stalk. So it was pretty simple and straightforward. He had a dominant trait and a recessive trait. Okay. Things are not always as clear cut as the Mendel Punnett squares. Okay, there's lots of times where there's multiple alleles that or multiple genes that affect a trait. There are times when the phenotypes are not just two. It's not just tall and short. It's tall and short and medium, or somewhere in between. So there's lots of different things that can happen. For example, in Gregor Mendel's mind, if you had a black mouse and a white mouse sit crossed, he would expect that there would be a mix of black mice and white mice in the offspring or in the F1 generation. He would never have even considered that there would be a gray mouse in the mix. But that's exactly what happens when we're talking about incomplete dominance. Okay, so he figured out that the offspring look like their parents. But he was completely unaware of this phenomenon we call incomplete dominance. Okay? Incomplete dominance is a cross between organisms with two different phenotypes producing offspring with a third phenotype that is a blending of the parental traits. So you can remember it this way. If you cross a red flower with a white flower, you get a pink flower because what does red and white make? It makes pink. So it's a mixture of the two. And in this type of dominance, we typically always use all capital letters because, in a way, all of the alleles are dominant because when you have the heterozygous version, like capital R, capital W, instead of red covering up the white, well, we see the white and the red, okay, because it's a mixture of the two. So the white and the red blend together to form a pink version for the heterozygous type. Okay, now these ones still have, the Punnett squares, you set them up exactly the same way. If it's a red flower, it's going to be capital R, capital R. If it's a white flower, capital W. And if it's pink, it's RW. And it's almost in a way a little bit simpler because there's no guessing. There's no two options of genotypes for any particular phenotype. It's just the dominant one one allele is red, one allele is white, and then the heterozygous one is always going to be the mixture of the two parental varieties. And then if you took two of the pink ones, the two of those offspring from the previous cross, and cross them together, you're going to end up with, you can have some white ones, you can have some red ones, because they still have those alleles in them, the R allele for red. So if both of them have an R allele and the offspring gets two R's, it's going to be red. Okay, so that's how that works. And so you want to think of incomplete as mixing, like mixing up paints. Red and white make pink. Red doesn't totally block or dominate the white. Instead, there is an incomplete dominance. So we end up with something in between the two. So it is a blending of the two. So helpful tips to recognize. So when you're looking at a problem, in number one, you see all capital letters, you know it's going to be either incomplete or codominance, and we'll talk about that in a minute. But you always want to look at that third phenotype. The parents are going to be each have one phenotype. What does the offspring look like? Is the offspring a mix of the other two? Then it's incomplete dominance if there's a mixing of the two. Okay. So our other option we were talking about was called codominance. Incomplete dominance, neither one of them show fully. so it's kind of incomplete. Codominance, co means together or cooperate, meaning they coexist. You have two colors, you see them both. You don't see a mixture, you see both colors. So in these examples, you see brown and you see white. And then these flower here, you see red and you see white. You don't see pink. You see spotted. Okay, so that's what codominance tends to be: things that are spotted or striped or mottled, or things like that. Okay, so it's very similar to incomplete dominance. The hybrid shows a third phenotype. The only difference is that the third phenotype is not a mixture. And it's more of something in the middle where you see spots. Okay, incomplete was the blending of the dominant and recessive traits, but in codominance, you see both of them together in the phenotype of the hybrid organism. For example, if you have a red and a white parental variety, then you have a red and white spotted offspring. Okay, so codominance is you see both, is like spotted. Incomplete dominance is where you see the blending. Okay, lots of times you're going to see codominance when we're talking about fur color and feather color. It happens a lot in those kinds of things. So with codominance, you have a cross between two different phenotypes, produces a third phenotype in which both of the parental traits appear together. So in this one, a white cow and a brown cow makes a white and a brown cow. So you see both. It's codominant. This is an example of a Punnett square. Using this, again, all capital letters. The black chicken would be BB, the white chicken would be WW, BW, the heterozygous version would be, you see both. And I know this chicken looks mostly black, but hopefully you can see that there are some white checkered feathers in there. So you see both. You see both white and black. It's not a mixing, so it's not a mixture. So this chicken isn't gray. It's you see them both. You still see white, you still see black, but it's just both on the same organism. Okay, the way you set these up are going to be identical to the Punnett squares you used before. You still need two parents, one parent on the top, one parent across the bottom, and you fill each square by pulling from the side and pulling from the type, and then you identify the genotypes and the phenotypes. Remember, genotypes are the letters, phenotypes is what it looks like. So in this case, checkered or black or white. Okay, so that is codominance and incomplete dominance. Again, here's an example of both of them. Codominant is where you have red and white and you both see red and white. That would be a codominant example. This is incomplete dominant where you have a red and white and you see pink. Okay, so that's the difference between the two. Incomplete is a blending. Codominant, you see both as spotted or stripes or mottled or something like that. Okay, bye guys.